Good morning, students. So I'll be talking about navigations, and also in the last class, we talked about some of the basics of the Linux, for example, path and what are the kind of directories and all that. So adding on to that, what we will do today is we will learn more about directories, what are some special directories are, and we will start with some of the data sets which may we will be using later on also. For example, RNA-seq data, as in if you look at the central dogma, then you have DNA, then you have RNA, and then you have proteins. So to keeping the theme of the structural and computational biology, we will be kind of working on files which are more of related to the biological data sets so that you are in the theme. One such forum is or portal is ENCODE project portal. As you see here, you just type here ENCODE project.org and this interface will open. And basically it hosts uh, lots of data sets like big data at the interface of let's say RNA sequencing data, genome accessibility data, and lots of things. So you can actually explore many things here. They have tabulated also here as per kind of a big umbrella programs kind of thing. So we'll start with some small files and we'll go about uh, exploring the commands that you use often to view file contents, and I will go into why these commands are important at times when you have to take a peek into the files. Okay, so first thing you do is you go to experiment search in the data section, and it will provide you this kind of an interface, which is very easy to kind of explore. They have many kind of layout, as you see here. We can just simply go into, for example, we are talking about total RNA-seq. So we will just do selection of total RNA-seq. And of course, you see here biosample, meaning that which organism you are interested in and what kind of cell line or tissue you are interested in. So here we can say one of the famous cell line that is used in kind of worldwide for hematopoietic cells or blood cells as per se is GM12878. Okay, so you select this and then you see 5 data sets are there, which is of the total RNA-seq. So what we do is simply we select here and we go down. You have more details in terms of like you see what are all the labels and everything are there. For example, Homo sapiens, GM12878, cell line, isogenic. Again, you have the name here. Then there are some more details which we are not so much interested in going about right now. Okay. When you go down, they basically show all the flow diagram, how they started from raw data to like final output file. But what we are right now interested in is just RNA sequencing results in terms of how much a particular gene is expressed in terms of numbers. Okay, so we will go to this file details tab, and you will see on the left side many kind of categories. As you see here, file format, big, and then TSV. So we are right now interested in this TSV file. So we'll click on this. So all these details, so many files were there, but then once you apply this filter, you see these 6 files. One thing to note here, if you're using this website, is keep track of the version of the genome that you are actually analyzing. Let's say back of the hand, you're working on some project, the data is from particular version of the human genome. Make sure it matches here also. Okay. So once we do this, you see these 6 files, and in some of them there'll be like a star, which is like, I guess, most downloaded file or highly used version of the all the sequence, all the RNA quantification data that you have. So you once you reach here, you can click on this download. But see, now we are going into computer programming, so we are not going to do left click. We'll just do right click here, okay, and we'll do copy link, okay. So once we do copy link, we go back to our terminal, okay. And what we will do right now here is, let me increase the font size of the screen. Now, to get that file at your desired folder, what we can simply do is, once we have copied the link, we can say, either in Ubuntu, you can say wget, okay, and just paste the link, okay. Once you do that, then press enter and it will work. In Linux, in Mac system, you need like this, curl command -jlo, okay. So since I have a Mac system, I'll just do this and press enter. So it is now downloading, okay. Great. So once this happens, you will be able to see this particular file, encff345shy.tsv. Now, TSV means tab separated values, meaning that this file likely is going to have header and then the data content, as you see normally in Excel sheet and all. But our goal for this session is to learn about how to view files on the screen, okay. As you would see somewhere in GUI, you double click on something and then it opens and something like that, some text file. But we are not going to do any double click. As you know, keyboard is our friend here. So we are looking for commands. One of the commands is called head, okay, meaning that look at the first few lines, or to be specific, look at the first 10 lines of the file that you are interested in. So as always, the first word is the command, and then you put a space, and then you say ENC. You don't need to type everything, you just press tab, okay. So what you see is, I can read it out here. See, it says gene ID, then transcript ID, length, effective length, expected count, ppm, fpkm, and all these values. And once the header is defined, then you have the values coming at every line, right? So it is giving you the first 10 entries, meaning that header plus 9 entries. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. Likewise, if you want to see the bottom of the file, back of the file, like back 10 lines or something like that. So you say tail. Okay. So head and tail. Tail, ENC. So then you're getting the last 10 lines of the file. Imagine if you want to see more than 10 lines. So let's say I don't know how to do that. So as I told you in the last class, you could go about looking at the manual. So what we do is MAN, then head. So what does it say? It says -n means count. Okay. So print count lines of each of the specified files. So what I learned here is I have to put -n, and also it says that each of the specified files, meaning that head is not limited to just single file. If you want to do multiple files, you can do it. Just put them as an argument one by one, like this, right? So I will say head minus n50, and then I will say enc. So now I'm getting 50 lines. Okay. So this is how you basically explore the different options that you have for a particular command, right? So likewise, I can say tail minus n50 enc. So I got 50 lines from the file. Okay. So this is one type, head, tail. Then another command is less. Okay. So this command, let me clear the screen. So you can clear the screen by pressing Control L. Okay. So you press Control L and then your screen will be cleared. So I say less and then enc. Okay. What it does is it does not just give you the file content and go back to the command prompt. Less is saying that, okay, I'll show you all the content of the file, but not all at once, but page by page. Okay. So for page by page, as in you're seeing the first page of the file as much as you can see it on the screen. Okay. And then you press space. So space bar. So it will go like it will pull one more page from the file and show it to here. Again, press spacebar, 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 spacebar. Okay. You can explore what all options I can go about kind of viewing the file by doing the manual of the less. Okay. You have many options that you can explore. Okay. Another command is again for viewing, it's more. Okay. So it's almost sort of does the same thing. People mostly use a less command, but again at your preference. Okay, so you have both serving as similar purposes. If there are any subtle differences, I am probably not aware of. Okay, the other command is called cat. Okay, so cat and then enc. What it does is it gives you everything from the file. Okay, right? The original purpose of the cat command was to bring couple of files together. Okay, means concatenate. Okay, so that's kind of a purpose of this thing. So if you say file 1, file 2, file 3 and all cat them all together, so let's say maybe I just make on the fly a couple of files. Okay, so I say A here. Okay, then vimp dot tsv. Say B here. Okay, and C dot tsv. Say C here. Okay, so what I can do now is say cat A B C. Okay, and all three will appear. And you can imagine lots of uses of this. Let's say you have to put couple of things in order, first, second, third. You can just say cat first, second, third, and then you can take them all and put it in another file or something like that. Okay, so this is about viewing. Okay, but there are many more things also awaiting in terms of, for example, like this. So cat abc dot tsv, right? But I want line numbers according to the like for each row which is missing right now, right? So I can say cat minus n a dot tsv, b dot tsv, c dot tsv. What is happening is here it will say it's the first because that's the first line in each of the file. Okay, but If there were two lines in a.tsv, then it will say 1a and then 2 whatever it was written there. Okay, so it basically enables line number in each file to show. Okay, likewise you can see the manual of cat and see many options that they have. Okay, see, for example, it says cat minus s squeeze multiple adjacent empty lines causing output to be single spaced. Okay, then you have cat minus n, we already explored. Okay, cat minus e display non-printing character and display the dollar sign at the end of each line. These are some certain uses of the cat command. Okay, I guess for viewing part, this should be good enough, and we will do more commands related things in a separate video where I'll go about looking at first couple of special files that are there which you don't see in terms of the file ID, and also a little bit more towards how to do some advanced uses of this navigation or viewing part. Okay, but before I go there, let's look at a couple of things in terms of the directory and file structure. Okay, so in the class we learned about absolute path, relative path, and all that. I'll just add a couple of more things. For example, whenever you create a new directory, it's, for example, I created this new directory, Linux command cheat sheet, like this. So I say ls. So I created all these files right now. You just saw it. Okay, but if I say ls minus a, meaning that show all the files or directories or folders, directory means folder, that exist here. So you will see two more things. One is dot and another one is dot dot. Okay, these two are special directories. Okay, meaning that this dot is saying that current directory. Okay, and dot dot is saying the parent directory, meaning in which directory this guy is residing. These two directories are critical to have everything connected. For example, if you want to go anywhere from this particular directory, how will you go on the command prompt? You don't have any ways to click or something like that. So in the class we learned, in the first class we learned about, I guess, cd command, right? So I said change directory. So how will you change if you don't have any link? So this dot dot is actually a linker. So if I say cd dot dot, meaning that go to my parent directory. Okay, so then I'm here, BEC computer programming. Let me clear the screen. Now here also I will have ls minus a. I will again see this dot dot. Okay, because it is essential to be connected, right? So when you create the directory, this comes with it. As I told you, like we were discussing in the class, when you create a file, the timestamp, time of creation, you know, all those information comes together, permissions and everything. So likewise, when you create a directory, these two has to be created. Okay, now if I want to go to again Linux command cheat sheet, I'll just say li and press tab and I'm there, right? The If you recall, in Windows system you have two things. One is, if you look at the left top corner, you have go up and go back, right? So go back. I want you guys to understand or explore the differences between go up and go back, and I will discuss that in the class. Okay, so for now the video, I'll pause this video, and I want you guys to, as an assignment, I want you guys to explore this ENCODE website and do the download for A549 cell line, which is a lung cell line. Okay, so A549. Do the downloads for the RNA-seq data, this TSV file for A549 cell line. Okay, thank you.